Hello, my name is Elise Spurling, and I'm a student researcher at Pittsford Mendon High School. My project is titled, Loss of Function Mutations in CSX27 Reveal that Poor Activity is Required for CRISPR-Cas13b Antiviral Defense. In general, bacteria use CRISPR-Cas systems to defend themselves against phages. In type 6b CRISPR systems, the effector enzyme Cas13b recognizes and cuts viral RNA. But these systems also have small accessory proteins like CSX27 that scientists think might help control what happens to the cell during infection. Earlier studies suggested that CSX27 might actually inhibit Cas13b, but these results came from deleting the CSX27 gene. Unfortunately, deleting a gene inside an operon changes transcription and often changes expression levels of neighboring genes. So, the field was left with a very important question. What does CSX27 actually do? My goal in this project was to answer that question directly. The main goal of my project was to determine whether CSX27's poor activity and not accidental transcriptional changes is essential for Cas13b antiviral defense. To do this, I needed a way to disrupt CSX27's function without disrupting the operon it sits in. I hypothesized that if I introduced structure-guided point mutations at residues predicted to line the pore, I could specifically disrupt pore activity while keeping Cas13b expression normal. If poor activity is required for defense, then those mutants should lose antiviral protection. If poor activity is not required, then mutants should behave just like wild-type CSX27. To test this, I used structural modeling tools, including AlphaFold and PyMol, to identify 4 residues positioned inside the predicted CSX27 pore, or at key oligomer interfaces. These were E8, E126, Q133, and R205. I mutated each residue to alanine, producing subtle changes that disrupt side chain interactions, but keep the overall protein and operon intact. I verified the constructs by PCR and Sanger sequencing, and expressed them in E. coli, along with Cas13b in the native operon. This ensured that Cas13b expression levels stayed constant between strains. Then, I measured antiviral defense using 2 assays. The first was a plaque assay, which we used to quantify plaque replication by counting PFU per milliliter. The second was growth curves, where I infected cultures at different MOIs and monitored optical density to see whether cells entered infection-dependent dormancy. Both assays together gave a clear picture of how the mutant behaved during infection. In plaque assays, the difference between the wild-type CSX27 and the mutants was very clear and very consistent. Wild-type CSX27 produced very low plaque numbers across all replicates, showing that the cells were effectively restricting phage replication. In contrast, every pore-disrupting mutant, E8A, E126A, Q133A, and R205A, showed high plaque counts, almost identical to the empty vector control. You could see this not only in the PFU numbers, but also in the plaque appearance. Mutants produced large, sharp-edged plaques typical of uncontrolled phage infection, while wild-type plates often had tiny or faint plaques. The only partial exception was R205A, which retained a small amount of defense. Its PFU levels were still much higher than the wild type, but consistently lower than the other mutants. Taken together, these results show very clearly that Cas13b alone cannot fully restrict phage infection, and that CSX27's poor activity is required for strong antiviral defense. Next, I looked at infection-dependent dormancy using growth curves. At MOI 0, all strains, wild-type mutants and controls, grew normally. This is important because it shows that mutations didn't affect baseline bacterial fitness. But once the cells were infected, the difference became obvious. Only the wild-type CSX27 strain entered strong, rapid dormancy. the growth curve flattened out very quickly, which is exactly what you'd expect when the defense system is fully active. All 4 poor mutants, however, continued growing almost as if no phage were present. Their curves rose smoothly, matching the empty vector control. R205 did show a slight slowdown, which lines up with its partial phenotype in the plaque assays, but it still failed to undergo true dormancy. So across both assays, the conclusion was the same. Disrupting CSX27's poor residues eliminates CSX27-dependent defense. These results demonstrate that Cas13b's RNA cleaving activity isn't enough on its own to protect the cell. To mount a full defense, the bacteria requires CSX27's poor activity to drive the dormancy response that stops phage replication. This helps resolve the confusion from earlier CSX27 deletion studies. Those deletions unintentionally disrupted operon structure and altered Cas13b expression, which made CSX27 appear inhibitory. But when the operon is preserved, and this means that only poor residues are changed, CSX27's real function becomes clear. It enhances antiviral defense, not inhibits it. The partial phenotype of R205A also suggests a true structure-function relationship, supporting the model that CSX27 forms an oligomeric membrane pore. This work is important for several reasons. First, it corrects a key misconception in the field and clarifies how this CRISPR system actually operates. Second, it highlights a broader theme across CRISPR biology. Effective antiviral defense often requires linking nucleic acid detection to a larger physiological response. Here, Cas13b handles RNA sensing, while CSX27 provides the membrane-based effector step that pushes the cell into dormancy. Finally, this helps build a more complete mechanistic model of type 6b CRISPR systems, which are still relatively understudied compared to systems like Cas9 and Cas12. Before I dive into future directions, I'd like to take a minute just to talk about some additional technical notes. So, to make sure that these conclusions were reliable, several important controls were used. The first was that all constructs preserved the native Cas13b C627 operon, avoiding transcriptional artifacts. Residues were also chosen based off structural modeling, not random selection, and all assays were repeated with biological replicates and showed consistent patterns. Finally, the graded phenotype of R205A ruled out non-specific effects and supported the poor model. Together, these controls make the functional conclusions strong and well supported. So there are several follow-up steps that could build on this work. One is expanding the mutation panel to map additional poor-facing residues. Another is using biochemical or structural approaches, like cryo-EM, which I'm actually currently doing now, to directly visualize the CSX27 pore. It would also be interesting to test CSX27 in different bacteria or with different phages to see how universal the mechanism is. Long-term, this system could be adopted for synthetic biology. For example, you could design engineered pores that activate under specific RNA signals. So, in summary, mutating pore-associated CSX27 residues removes antiviral defense. Mutants behave like the empty vector even though Cas13b is still expressed. Only wild-type CSX27 triggers strong infection-dependent dormancy. Therefore, pore activity is essential for Cas13b-mediated antiviral defense. This project clarifies CSX27's true role and shows how accessory proteins help convert RNA detection into a full cellular response. Thank you so much for listening, and I look forward to your questions.